Hey everybody, how's it going? Now I'm going to walk you through how to make your project of RNA. So we're going to be making a model of it, and this is super exciting. And here we go in theory. Okay, let me move this over. First of all, what I did is I made a piece of DNA, right? So I use Play-Doh because that's what the kid next door had. So I borrowed it to do this project. Um, and you can obviously use anything you want. Another time I did this, I made it out of macaroni. Um, so, and you'll see that in examples of this. So anyway, here you go. So I rolled a long thing of red Play-Doh, and this is going to be my backbone of my DNA. So first I'm doing my DNA, and then I'm showing you how to do that RNA. So this, this here is going to represent my sugar of my backbone. Okay, so then this is going to be, then I had to add my phosphate, right? Because remember I have phosphate, sugar. So phosphate deoxyribose. So I added these little rings here that you can see in yellow to be the phosphate. Okay, and in DNA, obviously it's double stranded, right? So I had to make another one. So you can see below here, it looks exactly the same. I have my, the red is representing my deoxyribose, and the yellow ones are my phosphate. Okay, so then I put in my nitrogen bases, right? And we know for DNA, A goes with T, C goes with G. So we're going to make this consistent, right? So everybody should do the same thing. So our top, if we laid out our DNA flat, the top one is going to be A, T, T, C, G, A. So that's what you're going to do, make those things. So I represented them in different colors, right? So I have this kind of bluish teal color is representing adenine or A. So you can see I have it on, whoops, I have it on the end there. And then I use this like peach color for thymine or the letter T. So I have two things of thymine here. I used white for cytosine, which is right here, and I use brown for guanine. And then I have a repeat of my adenine. So that's my one side of my DNA, right? So if I add that next thing, I'm going to have, I had to put the complement there, right? So now I have here A, and A goes with T, with thymine. This is going to be T, and T goes with adenine in DNA. T, adenine, or A, cytosine, guanine, so that's CG, and then GC, and then here I am again with A, and it's going with thymine, T. So in DNA, A goes with T, C goes with G. And you can obviously pause this video, and I do have a separate PowerPoint, which is just a PowerPoint, not a video, that you can refer to. Okay, so next, I got to make my labels, right? Because I'm big on labels. And so as you can see over here, I have DNA, A goes with T, C goes with G. And then I put here what I labeled it all, right? Because I used the red for my deoxyribose sugar. I used yellow for my phosphate. I used this blue for A or adenine. I used this kind of peachy color for thymine or T. Cytosine, I used the white. And then guanine, I used a brown. So this is kind of like my key, because you know how I like everything to be labeled. Okay, next. Okay, so now we're going to make the RNA model. Okay, so I have to have DNA in order to make RNA. All right, so in this model, if you look here, I have DNA, A goes to T and C goes to G. But it's different for RNA. In RNA, A goes with U, uracil. And then we still have C goes with G. So what we're going to do is we are going to make copies. We're going to make our RNA. It just really doesn't want to go, does it? We're going to make our this bottom portion of our DNA is going to be what we're going to base our RNA on. So remember in DNA, we have a couple different things from, RNA, we have a couple different things from DNA. And one of them is we have a ribose sugar instead of a deoxyribose. So RNA, that's where that R is getting from in the name. R comes from ribose. So DNA is the D for its sugar, deoxyribose. And RNA is R because it's a ribose sugar. Okay, so that's one difference. We have a different type of sugar. And the next thing is that we have uracil. We have this thing called uracil, and it's replacing thymine. So if you look at this key here, we have this, which mistakenly was written thymine, and we crossed it out because it's not in RNA. It's uracil is in RNA, not thymine. So we cross it out here and we put in uracil. All right. The other thing that's different between DNA and RNA is RNA has one strand. It only has one side. It's not like the double helix or the twisted ladder like the DNA. This only has one side. So what we're going to have to do is we make the copy, right, of this bottom one, and then we're going to make our RNA here. So I think that my next slide is a little more clear. Okay, so if I had separated out those two DNA models like here, see how I have the DNA together here? Well, what happens when it makes RNAs? It splits open. And then RNA is going to be made on top like here. So if I look here, this is my top part of my DNA, this is my bottom part of my DNA. And this is where I'm making my RNA. So I have the ribose sugar is going to be a mixture of this green and white here. So it's different than this, which is deoxy. So this is ribose sugar. And I have my phosphate still. That's the same. And now when I pair it, when I make it, I'm doing it out for the bottom one for DNA, right? I'm using that bottom strand to make this RNA. So what happens is I have this ribose sugar, my phosphates, and I'm going to match up those nitrogen bases. So remember, I'm making RNA. So here's my DNA here, right? So my first one is T. And this is the only confusing part I think there is about this. If I have this T, T goes with A. Okay, so I have A. Now I have this A down here, and this A, remember in RNA, this strand here, it cannot have any T's. So I'm not going to put T here, which in DNA, A always goes with T, but in RNA, A goes with U or uracil. So I have a A here, and in my DNA, I'm going to put a U because there are no T's allowed. So my next one is A, and then again, I have a U. And then here, this I have a G, and G goes with C, which is the same as DNA. Oh gosh. And down here, I have a C, and C goes with G, same as DNA. And then down here, I again have a T in my DNA, and that will match with an A in my RNA. So you can see I have both these models. And that's kind of how it happens. And then this RNA is going to float away, and the DNA is going to close back up like this up here. So so the DNA closes back up, and this was my right here, my RNA, and this is now going to go out of the nucleus. DNA can never leave the nucleus because it's too big, right? It has two strands, but RNA can leave because it's smaller. You know, just being single-stranded. Alrighty. So I hope this helps you make your models, and we should also look at. Sorry, let me move this up. We should also look. Make sure you look at the examples. Alrighty. Thank you.